Hi, micro folks. We are doing follow-up on our cabbage lactic acid fermentation experiment. So what I have to show you today is the actual cabbage. These are all aerobic um, batches. I have the cabbage for us to take a look at, and then I also have 24-hour incubated plates. Yeah, I took samples from each of the 3 aerobic cabbage batches yesterday, and I plated them onto TSA, our all-purpose media that will grow a wide range of bacteria and fungi. Unfortunately, I ran out of the Sabbro's dextrose media, so we're not going to be able to look specifically at our yeasts or molds, but the results, you guys, I think are just really cool, really fun. Okay, so you will remember we're doing these experiments trying to see what our ancestors knew about microbial processes, and we knew our ancestors, even before our ancestors knew about microbes, they observed microbial phenomena such as the rotting of food. So folks, this is our cabbage aerobic no salt. So this was our negative control. Sorry. Our negative control. So what would happen to the cabbage just if it was left without any kind of treatment? And you can see it's starting to brown. It's getting really kind of brown and yucky. Yucky, right? Very scientific. Slimy, right? Probably not something that if we had an alternative, we probably wouldn't eat this, right? So it's starting to decompose. And then if we take a look, oh, and also folks, the pH yesterday was pH 5, so the pH did decrease. And also when I smelled it, I felt like I could smell some lactic acid fermentation going on, but it might be that the spoilage decomposition microbes are in there fighting the good fight and maybe winning out, right? And so it was fascinating, folks, when I took juice from this batch and plated it yesterday. And this has just been incubated overnight. And again, you guys, I'm really trying to learn how to get the lighting right here. I moved into the garage because I found for morning shootings, shooting on the driveway, the light is really reflective. So I hope, folks, you can see this is the TS8 plate, our negative control cabbage. We have lots and lots of microbial growth there. It appears to me that we might have at least 2 different colony types. And what I also thought was fascinating, guys, is a gee whiz. I had plated some rotting lettuce. And this is the rotting lettuce on TSA. And I was like, gosh, you know, it looks to me that at least some of the colonies look somewhat similar. The rotting lettuce plate has been incubating, I think, 3 days. This our negative controlled cabbage has just been incubated overnight. So eventually these colonies will get bigger. But with the rotting cabbage, what we discovered is that when we plate to Sabbro's dextrose media, the rotting cabbage is sorry guys, the rotting lettuce was chock full of fungi, yeasts, and molds, and they are the great decomposers. So it is a shame for our negative control cabbage that looks like it's starting to rot that we didn't have the Sabbro's dextrose media to see if we could see some yeasts or molds there. We'll probably do this experiment again, tightening it up a little bit, and we'll definitely have Sabbro's dextrose media there. But again, what was really, really cool, you guys, was then if we take a look at this was our aerobic with salt. And folks, remember we said that the salt creates a hyperosmotic environment, so water moves from inside the plant cells to the outside. So by adding the salt, we created this liquid, and the liquid would help create at least a partial anaerobic environment because oxygen does not diffuse well through water. So even though we didn't seal the bag, right, air had access down here in the bottom where the cabbage is, at least the bottom layer, we probably did have an anaerobic environment. And what's interesting to me is at least down here in the bottom, where it is anaerobic, the cabbage is still looking good, right? It doesn't look like it's rotting. Up here at the top, where oxygen is present, it is starting to brown. It looks like it's starting to decompose. So yesterday, folks, this is after 5 days incubation, the pH was between 3 to 4. So it had started out at pH 7 and then had dropped to pH 4, and now it looks like it's continuing to drop. So I think we do have a lactic acid fermentation going on here. And again, if I open the bag, it smells like sauerkraut, right? That lovely lactic acid smell. And again, folks, this is very, very early on. This is the plate um, um, of the juice from the cabbage and salt, the one that we just looked at. And this is so hard to see, folks, but up here, up here, there's lots and lots of little tiny pinhead, no, or actually pinprick size colonies. And I think those might be the lactic acid bacteria. We're going to order some new media that will help support the growth of the lactic acid bacteria. They're a little bit finicky in their nutritional needs. So they are growing, but very slowly here on the TSA. But what's so fascinating, again, is if we compare it to our negative control, this is a negative control plate where there was no salt. We have lots of microbial growth here, and it looks like here most of those colonies, most of the microbes forming those big colonies, most of them have been inhibited, right? So I can see 1, 2, maybe 3 there, but it looks like we have maybe thousands of these little tiny, we're hoping, lactic acid bacteria. So that was very exciting to see by adding the salt, getting that little anaerobic area going, that we were able to inhibit one group of microbes, and we're hoping we're selecting for the growth of the lactic acid bacteria. And then the final one, folks, that I just find this so fascinating. Remember, we were testing the hypothesis, do you need to add the salt to help preserve the cabbage, or is it just creating an anaerobic environment? So this was another batch we did, and this is just the cabbage with excess water, no salt. And again, folks, the excess water creates that anaerobic environment through which oxygen doesn't diffuse well. So again, here we're just looking at this anaerobic environment, and the cabbage here, you guys, it looks the best of any of the batches, right? So it looks like the salt wasn't required. The salt maybe helped to create the anaerobic environment, right, by getting the net flow of water out of the cells. Salt will inhibit microbes too, right? So we can't discount that. But this cabbage looks the best of all. And the pH on this one, you guys, pH was pH 3. So it looks like it has maybe the lowest pH of any of the batches. And again, folks, just to me so cool, when we plate from the cabbage with excess water, we don't get any of those microbes forming the big colonies. And again, folks, I'm probably can't see this here. We'll definitely let these incubate a bit longer, and we'll look at the plates again. But again, I'm seeing thousands and thousands of these little tiny pinprick sized colonies, and I think those could be the lactic acid bacteria. So we might argue that it looks like the excess water did the best job at inhibiting the microbes that are growing so well in our negative control cabbage, right? So again, I'm hoping those are little lactic acid bacteria. Okay, folks, so this will probably do it for this experiment. I am going to be plating the anaerobic cabbages. We'll be looking at gram stains. But I think what we'll do is we'll probably repeat this experiment, and what we'll do is, as we go, we'll take serial dilution so we can actually estimate the concentration of the bacteria as we progress through the fermentation. But I think we've shown that just creating the anaerobic environment, it looks like that did select for the lactic acid bacteria. So I think that's just fascinating. Okay, I hope you guys can go out and eat some good sauerkraut or kimchi or some good pickles to celebrate this experiment. Okay, guys, see you in the next video.